couple quick announcements before we get started. What's happening next Tuesday at 8 a.m.? Final. Yay. Great excitement. 50 questions. Same deal as always. You know, multiple choice, etc. Big blue form. Any questions on the final other than what's going to be on it? Everything.
Is it worth the same as the midterm?
Yes, the same. It's worth exactly the same as all the midterms. So the 3 exams are worth exactly the same number of total points. As soon as I get your clicker answers on Friday, which we will have, I will be posting those clicker scores online. Other questions, comments, worries about the exam? Yeah. It is 8 a.m. on Tuesday. 8 a.m. Tuesday. 8 a.m. Tuesday, 8 a.m. Tuesday, 8 a.m. Tuesday, 8 a.m. Tuesday. And those people who have been taking the test in the testing center and can't get a time there, send me an email and we'll work something out. A couple other things. I got all of the audios for the lectures since the last midterm. They're uploaded as MP3s on D2L. So if people want the audio as well as the video, those are there. And I'll be doing that for this lecture and also for the review on Friday. Yeah?
So for the vote, I'm working really hard to push my grade forward. And I have a question about the clickers. So you said, I know you mentioned before that you wouldn't necessarily like hinder a student if their clickers weren't great, but are the clickers going to be graded like as they are, or are they too going to have a little bit of a curve, like if the whole class missed one, you know, you just throw that one out?
Okay, so the question is, so I'm going to paraphrase your question here, but what about clicker points and how I'm going to use those? I'm going to normalize them just like I do everything else, and I'll look and see if somebody like downloaded my clicker questions beforehand and knew all the answers beforehand. That'll be pretty obvious, and I'll cut that off. So yeah, I'll be doing it just the way I do the exams, exactly the same way, and those are just going to be 10%. So 30, 30, 30 for the 3 exams, as you were asking about, and then 10% for clickers, which adds up to 100. How convenient. Okay, another quick thing, the biology seminar. I know you've got other things on your minds right now, but tomorrow's biology seminar will be by Victor DeFilippis, who's a vaccine virus researcher from OHSU, so it'll be at noon in SRTC 247. I think that's it in terms of announcements for today. In the back, yeah. Audio for the guest lecture? Not that I know of. I'm guessing somebody might have it. If they do, please send it to me or post it on D2L. Yeah. You emailed it to me and it disappeared. Maybe it would be good if you emailed it to me then again, if you would, and I will post it. Any other questions, comments, worries, etc.? As always, what we get through today will be on the exam. What we don't get through today will not be on the exam, even though it's totally cool and awesome and amazing. But, you know, that's the nature of the beast. So let's get started with our clicker questions. Small regulatory RNAs that regulate translation generally bind the 3' UTR, the 5' UTR, cause RNA degradation, make about 20 base pairs with the target messenger RNA are derived from viruses.
They said microRNA is definitely not the same.
20, 15, 10, good idea. 5. Okay, so key to this question is really about regulation of translation. The regulation of translation is going to be your microRNAs. These are the ones which are going to be just binding to but not causing slicing to take place. And so they bind at the 3' UTR. Yay. Good. So our second question for today. The presence of a single band on STS page indicates that you have a single pure protein with a specific molecular mass, a pure heterodimeric protein with a specific aggregate molecular mass, a phosphorylated protein, a mixture of many proteins, or a DNA binding protein. One thing about clickers, it's just the last one you press that counts, so it's always worth voting. 15.
I think that's what he's talking about.
10. 5. Yes, it's always worth pushing the button. And yes, at least indicates that you have a single pure protein with a specific molecular mass. That's not always the case, and you can actually confuse yourself. You have to be careful about this sometimes, and we can talk more about that later, exactly what that means. Heterodimeric protein is usually going to give you 2 different bands because you've heated it up with STS and treated with beta-mercaptoethanol or something which is reducing your disulfide bonds. So usually that will give you 2 bands if they actually have different sizes. And that's why I put heterodimeric there. A phosphorylated protein could be the case, but not necessarily. You really have no idea whether it's phosphorylated or not. Hopefully it's not a mixture, because if you have all of these things with the same molecular mass, that would be a real problem. Again, that's partly why you can have issues with this. And a DNA binding protein, sure, it could be a DNA binding protein, but you really don't know.
For siRNA, isn't this S stand for small?
S does stand for small, yes, for siRNAs. And M stands for micro. So they're also basically 2 things which are small.
So the first one, the fact that it said small made it seem like it was specific.
Yeah, but you would say an siRNA. So there are lots of small RNAs. Sorry, the question is, I didn't repeat it here, but the question is about small RNAs. And small nucleolar RNAs are small RNAs, small nuclear RNAs. So small is unfortunately a really bad term. And siRNAs, you're completely right. It's small interfering RNA, but that's only one of the different small RNAs. So that's the difference there. Okay, so we'll continue today talking about first a couple of protein methods, just to finish off with the protein methods, and then move into nucleic acid methods, which, again, I'm not sure quite why they have this order in the textbook, but we're sticking with it, at least for now. We talked last time about STS page, and I just had that question, and basically your STS page is separating based on relative molecular mass. And so that's what you have here on this axis. What we have on this other axis is the last thing that I talked about on lecture on Monday, which is isoelectric focusing. So this is separating based on the isoelectric point of your protein. It's a much higher resolution technique, and if you happen to have a phosphorylated protein in a mixture with a non-phosphorylated protein, those will have different isoelectric points because it's a different charge. And so you can have one protein that is modified in a bunch of different ways. And it's quite probable that all of these spots here would represent one protein with a lot of different modifications that happen on it. And so this 2D gel electrophoresis is a wonderful way of separating out each of the individual proteins. And some of these gels are literally about this big, almost half a meter on each side. They're huge. But with that, you can basically separate every single protein that you have in a particular cell. And so each of those individual spots. And so that was until mass specs got really, really good. And even people still use them, ways of doing what they call proteomics. So looking at all of the proteins which are being made in any given cell at any given time. So these two-dimensional gels was one way of doing that. Yeah?
So we say it's a higher resolution. I was just curious if SDS page specifically referred to when you're trying to separate by molecular mass, or if that's a more general term and this is just isoelectric separation is a more specific subset of that.
Okay, so the question here is basically more on terminology than anything else. And so this particular, the two dimensions here, the first dimension is isoelectric focusing, and the second dimension is SDS page. And so you're separating in two different ways here. So separating based on the isoelectric point and separating based on the relative molecular mass. Yeah?
How do you get the gradient for the isoelectric?
How do you get a nice stable pH gradient?
Yeah.
As far as we're concerned for this course, magic. But yeah, there are ways to do that, but usually you end up actually buying something which is already set up. So yeah, it's basically magic. So one question, of course, with this is, what the heck are each and every one of these individual spots? And if you are lucky enough to have an antibody, that's a really great way of figuring out what each of these spots are. So a Western blot is just taking this two-dimensional gel here, a god-awful mess, and transferring it to a solid surface. Usually it's going to be some kind of membrane. And then you use your antibody, which you've bought from somebody or made because you put some antibodies or made some antigens and put them into a poor little bunny rabbit or something like that. And then they make antibodies. You use those antibodies. But those antibodies are going to be very specific. And in this particular case, this is almost definitely something that someone bought, which was a anti-phosphothreonine. And what this does is this will bind to only those proteins that are phosphorylated on threonine residues. And you see there are actually relatively few of them here compared to all of the proteins that you had up here. And so this is a really nice way of identifying the protein that you're looking for, or looking for specific modifications, something like that. So it's the transfer to a membrane, incubating with antibody, and then you have to have some way of detecting that antibody. And the easiest way to think about that is you actually have a radioactive antibody, and then use film, and that radioactive antibody will express on the film. There are other ways of doing this as well that we'll talk about when we talk about DNA sequencing in a couple of minutes. So I mentioned 2D gels. They're great. They're wonderful. You get this amazing resolution. They're a pain in the neck to make, partly because of the magic of isoelectric focusing that we just talked about here. So getting a 2D gel and making it reproducibly is extremely difficult. So, and if one of those spots moves even a tiny little bit, it's going to be very hard to identify what that was in the first place. So what people are moving more and more towards now is mass spectrometry, mostly because these mass spectrometers have gotten much, much, much more accurate. And that process is basically, for a single protein, is you take this protein, you chop it up into little pieces, usually using a specific protease. In this case, it's trypsin, which cuts after specific amino acid residues that are not important as far as this course is concerned. And that will give you short peptides. And then you can put these peptides into a mass spectrometer, and they will tell you exactly, and when I say exactly, it's to like about 4 decimal points, the molecular mass of each of these peptides. by knowing what the sequence is of the genome of the organism that you're looking at, by doing all the DNA sequencing stuff, again, that we'll talk about in just a second, you can now take these peptides and identify exactly what gene they came from in the genome. And this is done all, in the computer. You get a set of peptides and you say, hey, these peptides have to come from this one particular protein. And again, it's because you have this really amazing accuracy on the mass spectrometry. And it's gotten so good that you don't just have to have a single protein up here. You can take all the proteins in the genome and chop them up, throw them into your mass spectrometer, and then get all of those peptides which are being made in that particular cell at that particular time. And you can identify each and every single one of those proteins. It's really pretty mind-blowing how well this works. And it's a much more reproducible method of looking at all the proteins which are present in a given cell at any given time. Yeah?
So does that mean that the genomes are like on an open public database that basically any computer can just download their info to?
So the question is, does it mean that all of the data are available so you can go and search all of your peptides against them? And the answer is, for the most part, yes. There are going to be a few which are not, but I would say 99.9% of the genomes are on public database servers. One of the big problems that is happening now is those data sets are so ridiculously large that it actually has crashed our network a couple of times here when I tried to download them. So we're talking not terabytes worth of data, but petabytes worth of data in terms of what all the sequence data is out there. So it becomes a real computational problem. It's not an experimental problem. It's a computational problem right now. Yeah?
If you're doing mass spec with all of the proteins in a given cell,
Yeah.
and it's broken down into each peptide, how does it know which peptides associate into particular proteins?
So the question basically is how do you put each peptide into a particular protein? So you're exactly right. How do you do this? And how do you say, okay, this peptide belongs to that protein, the other peptide belongs to another protein? So the proteomics approach here is great if you have some way of separating things. And so what you have, what I ignored to mention, but now you bring it up, I can't sweep it under the rug, is you usually have some other separation technique that you use beforehand. And almost always this is going to be some kind of liquid chromatography, a lot like what we talked about last time. And so each of the individual peaks that's coming off there. And quite often, people will also do this with spots off of one of these 2D gels. But the liquid chromatography is a much more repeatable mechanism to be able to do that. The other thing that you can do, which is what's shown on the right-hand side of this slide here, is of those individual peptides, which will have a particular mass, you can do what's called MS-MS, so mass spectrometry on each of those separated peptides. And that way you can break each of those peptides down into individual sequences. And if you're lucky enough or you have a low enough diversity proteome, you can just use MS-MS to identify those peptides. Because what happens is, to get back to your question, how do you know what peptide belongs to what protein? If you have a specific sequence that then you can correlate with your genome, that's going to be specific relative to the genome. So you can actually do this without having that separation in the first place, which again is pretty amazing. But then if you are going to do that, you do have to have this MS-MS process where you get the sequence of each of those individual peptides, or at least some of the peptides that you think are going to be important. Okay, so now you've identified the proteins. That's great. You have the sequence of them, but it doesn't really tell you that much about how they work. And one great way to at least get an idea how they might work is to get a three-dimensional structure. You think 2D gels are hard. Getting three-dimensional structures is even more challenging. So we've got thousands, if not tens of thousands, of whole genomes, which are going to have anywhere from 1,000 or a couple thousand genes to 20-some thousand genes. So we're talking millions of genes with sequences to them on the order of a couple of thousand of high-resolution protein structures. So part of the reason for that, it's actually kind of hard to get enough of the particular protein, or for that matter, nucleic acid, that you need to make a crystal. And most proteins don't form nice crystals. And so that's the whole making of this crystal in the first place. This is the really hard part of this process, and very much an art and luck form. But if you do have a protein crystal, then you can do X-ray diffraction on this crystal, assuming it's a good crystal. And this is basically just physics. If you've got everything lined up in exactly the same way in your crystal, then if you have the beam that gets diffracted off from one thing in the crystal, it'll get diffracted in the same way by the next unit in the crystal, and so on and so forth. And what that'll end up doing is giving you this sort of Rorschach blot of different spots. And then through the magic of computers, you can go from this two-dimensional image to a three-dimensional image, which is what we see all these pretty pictures of in the textbooks. And so this is just one of those three-dimensional structures, which was gotten from X-ray crystallography. Again, the big problem here is making the crystals. You can make a crystal, great, wonderful, but I think of the virus proteins that we've tried to do, about 50 of them, we've gotten about 10. So it's doable, but it's a bit of a pain. Another way to do this is NMR spectroscopy. And NMR spectroscopy is wonderful if you have a relatively small protein and it's very soluble at high concentrations, much higher concentrations than you would normally have in the cell. And the reason for that is what you're looking for is NMR. Anybody, NMR, spectroscopy, chemistry, analytical chemistry? Yeah, so you're basically just looking at how atoms are related to each other in space. And each of the little dots on this particular plot represents distances between individual atoms. And again, lots of computation. You basically have a humongous table of distances that says, this hydrogen atom here is certain distance away from that, certain distance away from this one, et cetera, et cetera, et cetera. And then from that massive table, you put together a collection of three-dimensional structures that fit your data. And hopefully, they'll converge, i.e. they'll be mostly similar in most of your protein and give you a bit of a structure like this. There are a couple of really neat things about NMR because you're just doing this in solution. If you've got some areas where they're not convergent, you see this area right down here where things seem to be wobbling around a lot, well, that's probably because they actually are wobbling around a lot in the actual protein. And so this gives you some dynamics. If you have a crystal, it's crystallized. That's it. It's solid phase. And so here, this gives you information on those things. Limited in terms of size and limited in terms of solubility. So this is great if the things work that way. Yeah?
If they have some wobble, do you assume that little section is a matrix?
If they have some wobble, you assume what? Sorry.
Matrix versus domain. If matrix, if it has some flexibility in that.
Oh, so if there's some flexibility, what do you assume about that particular piece? Well, there are lots of potential reasons you would have flexibility. One would be it's normally associating with something else. Could be another protein, something else which is in the cell. Or it may actually be unstructured because that's part of its function. And again, when pre-Dr. Iverson talked about this way back when, right at the beginning of the class, some proteins are unstructured, and that's part of their function. So yeah, there are lots of potential reasons why you'll have a region which seems to be floppy in terms of this. Okay, so you can only get usually a certain number of protein structures. And again, it's on the order of about 1,000 right now. And that's been hugely advanced in the last couple of years. It was a couple of hundred, and now we're at a couple of thousand. So the main way that you usually are going to try and figure out what's going on with your favorite new protein is to go and look at the databases that we just talked about. So you have a sequence of your favorite protein, and then you compare it to all the other sequences that are out there, and you hope that somebody else has actually done something with a similar sequence, which, if you're really lucky, has a structure, and if you're even luckier, it has a structure and a function. And so that's basically the idea here. This is just one way of doing. It's a very simple way and completely open, funded mostly by your tax dollars, way of searching databases. It's what's called a basic local alignment search tool. I would say 95% of the people who know this don't even know what that acronym stands for. They just talk about blasting. They take it as a verb. Absolutely horrible. So you use this tool, and all it is, it's a web-based tool. So you put your sequence in, you hit go, and then, depending on how many other people are searching for their sequences, in a few seconds to a few minutes later, it's searched through these petabytes of data and comes up with something that is similar, or it says, sorry, there's nothing else there, which is really cool, but really frustrating if you've got something which is new. So, but this is a nice example of where we have our sequence that we put in at the top. This is our query sequence, and then what the computer has found here. And this is really similar. Everything which is in yellow with a letter here is an identical amino acid. Everything which is plus is a similar amino acid according to the computer. So we've got a phenylalanine and a tyrosine, a glutamine and a glutamate. You could argue whether these are similar, but one thing that should be pretty obvious here, lots of yellow. And one of the great things that this tool does is it gives you what's called an E number or an expect number. This is a probability of how often you would have this match by chance. And e to the minus 111 is a pretty small number. Usually the cutoff is e to the minus 3, e to the minus 5, depending on how you're looking at it. So this kind of similarity won't have happened by chance. It's something which is, I don't know, we'll see, certainly possible, but extremely unlikely this is happening by chance. So if it turns out that somebody knows something about this subject that you find, now you can make some predictions about what your favorite protein does. And then, of course, you can test some of those predictions in terms of looking at them. So that's all I have for protein methods. This is basically what we've talked about already, with the exception down here of the protein-protein interactions that we're probably not going to get to at the end today. So now let's talk about DNA methods. How many people are taking the recombinant DNA next term? This would be a fabulous course. It's basically going to take what I'm talking about in the next half an hour and expand it to 10 weeks. So it's really revolutionized what we know about biology, founded a multi-billion-dollar industry, all about moving DNA around. So we'll talk about a couple things: hybridization, blots, but mostly about DNA cloning. We talked about nuclear cloning, which is, taking the genome of one organism and putting it into another one. This is just little pieces of that genome, library sequencing, and PCR. Why should we care? Because I'd probably bet a case of beer that everyone in this room at some point will at least have access to their whole genome sequence. That's because of this kind of technology. And any of you who are moving into the medical profession, you'll have access to the genome of your patients. So what do you do with that? Well, that's a whole different question and story. Genetic modification, gene therapy. This is exactly how gene therapy happens. Drug production, I would say, of the pharmaceuticals on the market, particularly a lot of the anti-cancer drugs, 95% of them are made this way. The other ones are made purely synthetically. And the whole biotechnology research and this, et cetera, could go on forever, more or less. So what are we going to talk about? gel electrophoresis. This is a slightly different than the SDS page that we talked about, but basically the same idea, separating based on charge and size. Nucleic acids are great because they have uniform charge density. Makes it a lot easier. Oh, that was interesting. Probes, blots, and then recombinant DNA, how you put this stuff together. This is basically what they use as an outline in the text. All you need to do is chop up the DNA, put it back together, and make a whole bunch of it, figure out what you have, make some, and then figure out what that sequence is. So we'll go through each of those here as we move forward. The first of these depends on restriction endonucleases, which came from the study of viruses, exactly. So particularly the resistance of bacteria to infection by viruses. And what people figured out was that bacteria that were resistant to infection encoded these restriction endonucleases. They restricted the host range of these individual viruses. What these restriction enzymes do, they're endonucleases, so they chop DNA. And they chop DNA at very specific sequences, which is great if your virus happens to have that particular sequence in it. It's not so good if your genome happens to have that sequence in it as well. So how the organisms here dealt with this is they methylate that sequence in their own genome, and most of the viruses don't have methylated DNA that comes in. We already talked about DNA methylation in bacteria, which is important for replication origins, knowing what's old DNA, what's new DNA. So these methylation processes happen really quite frequently. So this is a way of protecting against virus defense, and almost always it's methylation. So methylation at adenines, methylation at cytosines. And all that does is blocks that restriction endonuclease from binding and cutting the host DNA, whereas it will chop up the virus DNA, or for that matter, and this is why it's really important for biotechnology, any DNA that's not been methylated. Yeah?
Do we see viruses methylating their genome?
Do we see viruses methylating their own genomes? Because it's an arms race between the viruses and this. So the answer is yes, and also some viruses avoid having these sequences. They seem to have evolved away from having these particular sequences in them. So, but one of the, again, this whole arms race, what it means is different bacteria have different restriction endonucleases for different viruses which are going to be potentially infecting them. And it turns out that there's almost all of the possible sequences you could think of in terms of a restriction endonuclease that will cut at a specific sequence. Yeah?
You said that bacteria have different restriction endonucleases for different viruses.
Mm-hmm.
Is that kind of like a one-to-one ratio where you have one type of antibody for one antigen?
Okay, so the question is in terms of restriction endonucleases, how many restriction endonucleases have versus how many viruses are infecting. And that seems to be really variable. So it's not like you have one restriction endonuclease and one particular antigen that's interacting with. So it's one restriction endonuclease that hopefully protects you against lots of different viruses. And so that's the idea here. And if you just think about E. coli, and if you look at restriction endonucleases, and if you've worked in labs, probably know all about restriction endonucleases, but the first three letters of any restriction endonuclease represent the species of organism this comes from. So here, EcoR1 comes from E. coli. And in fact, EcoR5 also comes from E. coli. So there are two different restriction endonucleases that you find in E. coli. And it varies from organism to organism. Some have huge numbers, some have very few. But many of them cut these different sequences, which is, again, extremely useful. They almost always cut in palindromic sequences, and a palindromic sequence is just the same sequence from 5' to 3' on both strands. So here, GTTAAC, GTTAAC, GAATTC, GAATTC. So same sequence on opposite strands, and one of the things that should hopefully be telling is that these things are probably also binding to half sites, like we talked about for DNA binding proteins, that are then bound by dimers, which will then bind to, and then since they're endonucleases, will end up cutting that DNA. And that's true for, I would say, 90% of these. A few of these restriction endonucleases have 8 base pairs that they bind to and cut, but most of them are going to be 6, 3 half sites for each of those. Yeah?
I'm sorry, I'm not following this half site concept. So, in the dimers.
Okay, the half site and the dimers. So this is great review, so I won't have to talk about it on Friday. So the idea is that if you think about DNA binding and your classic helix turn helix, let's take a helix turn helix motif from a bacterial DNA binding protein. So that single alpha helix is going to sit down in the major groove of DNA, and it's going to interact with the base pairs which are present in that major groove. One alpha helix, a couple of beta strands, are usually going to interact with 3 or 4 individual base pairs. 3 or 4 individual base pairs is not enough to give you binding specificity for a whole genome. On the other hand, 6 is a lot better. Still not going to give you for the whole genome, but a lot better in terms of your specificity. And the way you can get 6 is by taking that same protein with that same alpha helix that fits into the major groove and binds these 3 bases. You can put it together with another one that now binds to 3 base pairs in the major groove of DNA. And if they're in an opposite orientation to each other, they fit together through your little dimerization motif, which are leucine zippers, helix loop helices, or a bunch of other different kinds of dimerization motifs as well. Those now can bind to 6 base pairs. Does that make more sense? So this side binds to a half site, that side binds to a half site. Great review for that lecture way back when, depths of time, like 2.5 weeks ago, which will be on the final. So bind to and cut these sequences. This is great for protecting yourself against viral defense, protecting yourself against virus attack, I should say, and also critical for recombinant DNA, because what you can do is take any piece of DNA and cut it with this restriction endonuclease, and in many of these cases, you're going to end up with what are called these overhangs. So once it's cut, you're going to have a double-stranded break, but it'll have 3, 4, sometimes even more bases which are single-stranded at either end here. And you'll see that's really critical in terms of that next step I mentioned, which is ligation. So you can cut any DNA with these restriction endonucleases, put those DNA together, and they will get ligated to each other. So it's because all cellular life uses DNA as genetic material, you can cut it, you can put it back together. Yeah? An analog to restriction endonucleases in eukaryotes? No, there's no analog to restriction endonucleases in eukaryotes. So bacteria rule. Yeah? So the question at the back is, can you use bacterial endonucleases in eukaryotic cells? The answer is yes, you can. They don't seem to have evolved in those systems, but you can use them. It's rather dangerous to be chopping up your DNA here, and you don't have the same kind of specific methylation of those sequences in the eukaryotic genome. So if you're putting in an endonuclease, you'll be chopping up the cellular DNA, which, if you really want to, is maybe a good thing. But also, remember, these are 6 base sequences. So every 256 base pairs on average, they're going to be cutting cutting your DNA. That then is a real problem if you've got a ginormous genome. If you've got smaller genomes, it's a lot less of an issue. Yeah?
If these will religate with other DNA strands, why don't they just religate back together?
Why won't they religate back together? And the answer is they will. But usually you've separated them from each other using gel electrophoresis. Thanks for the transition. Greatly appreciate it. Yeah, you'll separate these things from each other. The other thing that you usually do, turns out that they need phosphates at the end for your ligase to work. And so if you get rid of the phosphate after you've done your restriction endonuclease, then those ends won't ligate back together. If you put in something that's phosphorylated, then that will preferentially ligate to that. So yeah, one way to separate them is on gel electrophoresis. We'll start here actually looking at the middle, because that's usually the way that you're going to separate the DNA, which you've chopped up with a restriction endonuclease. This is agarose gel electrophoresis. Separates between about 500 and 10,000 nucleotides. Here's just the 1,000 nucleotides here, 6,000 nucleotides at the top. This is great.